So good afternoon all. Welcome to today's Eawag seminar, which is organized in Zoom only. I am Daniel Odermatt. I'm a group tenure track group leader in the SURF department, and I coordinate the activities of the tenure track cohort. And since last year, successfully tenured group leaders have the opportunity to present their achievements of their tenure track period in a seminar talk. Our speaker today is the newest former member of the cohort, Joaquin Jimenez-Martinez. Joaquin is a group leader in the Department of Water Resources and Drinking Water, and his tenure appointment was completed in May last year. He leads the Subsurface Environmental Processes Research Group, SEP, as a research scientist at Eawag, and he's a lecturer at the Department of Civil and Environmental Engineering at ETH Zurich. The SEP group focuses on the study of chemical transport and reactions in biological processes in porous and fractured media, particularly in multi-phase systems. The group tackles these problems mainly by experiments performed over scales ranging from micromodels to field scale and blending them with theory and modeling. Joaquin obtained the bachelor degree in geology from the University of Granada and the master in geotechnical engineering from the Technical University of Barcelona. In 2010, he obtained a PhD in subsurface hydrology, again at the Technical University of Barcelona. Joaquin strengthened his research formation as a postdoc at the University of Rennes in France in 2010 to 2012, and was awarded an extension at the university's International Chair on Environment and Innovation until 2014. In 2014, he became a research associate at the Los Alamos National Laboratory in the US for 2 years. And in 2016, Joaquin started his tenure track position at Eawag and ETH, which he will now wrap up with this seminar talk. It is titled Multi-Scale Problems in Subsurface Environments, What We Can Learn from Each Scale. Please be reminded that we ask the participants to mute their mics. You can ask questions either in the chat window at any time or by raising your hands in the end at the question and answer session, where we will also repeat the questions that were asked in the chat. So please, Joaquin, the screens are yours.
Thank you, Daniel, for the kind introduction. Thank you also for to the organizers for giving me the opportunity to show part of the work that we do. Of course, it's impossible to summarize in 40 minutes the work that has been done in 5 years. That's why I decided to select a topic, which is the one that I'm going to address today. And I will do my best to take you in a journey that will cover different scales or a very similar problem at different scales. As Daniel pointed out, the main environmental compartment in which we work is the subsurface. And let me define what I mean by subsurface. First, we mainly focus in the unsaturated zone, which is consisting about the soil and the vadose zone. Vadose zone is a very American term that is used for the space that we have below the roots and above the groundwater level. And of course, the unsaturated zone plays a key role with many functions in the health critical zone. For example, in the biomass production, carbon storage. One of the main fluxes that we have about CO2 is the ones that happen from the soil into the atmosphere. Is of course one of the main actors in the water cycle with the water storage and transmission that happen between the soil surface and the aquifers. Is important in the transformation of nutrients, how we're going to see in one of the examples today, and is one of the main hosts or the main host of life in the non-universe actually. Then below that, what we have is the groundwater, is the fully saturated part of the subsurface, which also has some function in the critical zone. Groundwater itself is an ecosystem and play a key role in weathering minerals, in producing especially cations like sodium, potassium, magnesium, and transporting them. Of course, groundwater is important for the base flow of streams, but it's also very important sustaining other ecosystems, like for example, wetlands, estuaries, lakes, as I mentioned, rivers, or the transpiration of phreatophytes plants. So now that we know that we can divide the subsurface in at least two small subcompartments, I would like to point out that the subsurface is is a multi-scale research challenge, and we can think in this sense for many other environmental compartments, but this is very, very remarkable in the case of the subsurface because of the topological complexity. In the subsurface, we can have processes that happen or are relevant at what we call porous small scale, with, for example, a lot of microbial activity. We have other processes that happen at intermediate scale and are only visible or measurable at this intermediate scale. And then we have processes that only can be seen, can be addressed at the large field scale. However, something that I would like to point out from this slide is almost what is the slogan of the group, and is that the small scale processes control large scale observations. I believe, or we believe, that the integral, the convolution of many small processes is actually what we see at large scale. So in this multi-scale research challenge, what we have is also another challenge, which is we need a multidisciplinary approach to address the key questions. And for that, I would like to show you an example. If we think in the subsurface, like for example, soil, in which we have solid grains, here in brown color, we have fresh water, in blue color, we have air in black color, and we have a contaminant or a nutrient that is moving through this complex system. What we see is that the transport of this chemical is pretty complex and is pretty difficult to predict. Then on the top of that, we can have also microbial activity, microbial behavior. We can have colonies and biofilms. Of course, we can have planktonic organisms. We can have the action of the roots. And as you can imagine, there is a permanent feedback between these physicochemical processes and these microbial processes. So everything together invite to combine something that sounds very trivial, but not necessarily is done or often is ignored, to combine expert on physical processes, expert on chemical processes, and expert on microbiological processes. So, as I mentioned, my idea today is to take you in a journey in which we're going to cover different scales, but with a common thread. And the common thread is the surface reaction adsorption, which is widely known, widely studied at Eawag. And that will be the main point or link between the different scales that I'm going to show you with three different examples. Let's start with the largest scale, and with this example, this is what we call field scale. In this case, it's a project that has been recently closed and that we have done in Valencia, in east of Spain. In this case, the main goal was to assess the impact of irrigation modernization on aquifer recharge and the loss of fertilizer to the aquifer. And what we see here is a picture of this region. It's a very beautiful region with all these orchards, orange trees. And we can see here, for example, this exerts, this dam is 700 years old, actually from the Arabs, and it's still there and it's still working. It's quite impressive. So for those of you that are not familiar with what I mean by irrigation modernization, the irrigation modernization has been the conversion from flat on the left-hand side to the drip irrigation. The region is still converting. This is happening since the end of 90s. And this is how the conversion happened. In blue color, here in this animation, what we have is the irrigation sector, the group of farmers that decided to keep or being with the flat irrigation method. And in green color are the ones, and you have here time on the upper right corner, the sectors that has been converted into drip irrigation. So this region, as I say, in Valencia, and the aquifer below this agricultural area is in contact with this wetland and also with the famous rice that is used for paella. So people is very concerned that the agriculture that is practiced in this area could have any impact either in the albufera and also in the rice plantations. So I mentioned that the conversion was from flat, or is still happening, from flat to drip irrigation. And to characterize or to assess what has been the impact of this progressive modernization, what we have done is to use a numerical tool, in this case a bucket type model, in which we have considered field observations with the different irrigation schedule that you have in the case of drip and in the case of flat. And here what we have is daily values of in green color for drip irrigation and in blue color for flat irrigation. The same information was able to be obtained from stakeholder interviews about the fertilization. We were able to identify that there are 5 different strategies or patterns of fertilization. As I mentioned, we use a bucket type model in which our main target or our main variables, or the ones that we wanted to basically quantify accurately, it was the percolation of the aquifer recharge and the leaking or the loss of nitrogen to the aquifer. So, and these are the main results, or one of the main results of this project. The main take-home message from this slide is that drip irrigation has expected, of course, reduce the aquifer recharge and the nitrogen leak. But, of course, there are many buts. Because the aim of this project was to assess if the irrigation modernization was a good choice in a climate change context. And also, in particular, in this Mediterranean region, which with a type of precipitation. In this region, precipitation happens usually in spring and in fall, but in very few hours, you can have a lot of liter per square meter. And this is not the best, probably for an irrigation modernization. But let me show you or explain the results in details. On the left-hand side, what we have is the aquifer recharge. And what we see in blue color is the aquifer recharge that is produced by flat irrigation. And in green color, what we have is the aquifer recharge that is produced by the drip irrigation. As we can see, of course, drip irrigation has improved because in spring doesn't produce any recharge, but is still producing some recharge in fall. And now if I move to the right-hand side of the slide, to talk about the nitrogen leaking, as I mentioned before, we have been able to identify 5 different patterns or strategies of fertilization. 2 for the case of flat irrigation. This number means the kilogram of nitrogen, per hectare and per year. And in the case of drip irrigation, we were able to identify 3 different. Okay, and this is what we plot here with different lines, but in general, we can talk about blue tones and green tones, flat and drip irrigation respectively. And in the case of flat, we can see that there is an important input of nitrogen into the aquifer during spring, and also a little one in fall. But what it was our surprise is this huge amount of nitrogen that is still leaking to the aquifer, even if we are operating with drip irrigation. So yeah, in general, drip irrigation reduces the aquifer recharge and nitrogen leaking, but not as much as expected. And there are other drawbacks of drip irrigation. And the other drawback is the higher sensitivity to climate conditions. What I mean by that, in the left-hand side, we have the aquifer recharge versus precipitation for the different irrigation sector and for our 50 years of simulation. And what we can see is that, of course, the recharge that is produced by the blue dots, in this case by the flat irrigation, is higher than the ones that we get from drip irrigation sectors. But what we can see here is that the slope of this is different. So it means that when we have a wet year, okay, when we have a lot of precipitation, again, this precipitation happens in very few hours, then at the end, the performance of drip irrigation is not that different than the ones of flat irrigation. And this is even more remarkable in the case of nitrogen leaking. What we can see here is, again, in blue tone, what we get from flat. This is the nitrogen leaking in the y-axis, and this is versus precipitation and irrigation. The green show for the different drip irrigation fertilization strategies. And clearly, what we see again is that when we are in wet years, when we combine precipitation plus irrigation, usually irrigation is almost the same every year. So what is changed is precipitation. When we have very wet years, at the end, the performance of drip irrigation is not much better than the one of flat. And the reason behind of that is that drip irrigation, even if it's very local, make the irrigation itself much more efficient, make the soil keep a degree of saturation, a water content, which is relatively high all over the year. So when this type of precipitation, intensive precipitation, like a storm, in which you can have even 60, 70, 80 liters per square meter in 1 hour, then the soil doesn't have any capacity to storage water and even less to storage any nitrogen that is already there. So basically what we can see is that these very intensive precipitation events produce these pulses of nitrogen leaking into the aquifer. And of course, when you are using a numerical tool, like it was here the case, you need to do a sensitivity analysis for the parameters of your model. And in this case, what I show here on the left-hand side in this figure is the sensitivity of the nitrogen leaking to parameters included in the nitrogen cycle with the bold colors and with the empty color for the water balance or hydrological cycle. Again, in blue color, we have flat, and in green color, we have drip irrigation. And we have made a distinction between dry, mean, and wet year. We have selected 3 years, and in these box plots, what you have is all the irrigation sectors, and also the 50 years of simulation. So, as expected, of course, the parameters of the usage for water balance in our numerical model, of course, we can have a lot of sensitivity, and the results that we can get in terms of nitrogen leaking can change a lot depending on the set of parameters that we choose. But it's not negligible also the variability that we have, or how sensitive is the leaking of nitrogen to the parameter of usage to solve the nitrogen side. So after to get this, and we can see that it's different between flat and drip irrigation, and of course drip irrigation has expected is performing better than flat. What we decided is to do an analysis of parameter importance. And for that, what we did is to do a experiment rank correlation analysis in which we compare parameter values versus nitrogen leaking. And to our surprise, there are many different processes that are included in our model, either for flat and for drip irrigation, like mineralization, nitrification. But to our surprise, our model, it was very sensitive to the parameters that are related with the absorption, in this case with the partition coefficient. Absorption is very important, for example, for ammonia. So, and then at this point, we can ask ourselves, okay, but why? Why the main or the most sensitive part of our model is the one that is related with the absorption process? Then for that, we need to go one scale down, and this is what we addressed in this second example. Now we're going to talk about the intermediate scale. This is another project that we developed in the last year in south of Spain, in Cádiz, in which the soils, they are irrigated with treated wastewater, and also they are amended with the sludge. So what we realize is that because of the sludge and because of the use of treated wastewater, there was a high content of surfactant. In this case, we were mainly focused on the structural control of non-ionic surfactants in these agricultural soils. So As I say, we were able to identify many different surfactants like the alcohol ethoxysulfates, the nonylphenol polyethoxylates. The names are very exotic, but in this case, we were mainly focused on these alcohol polyethoxylates, which are non-ionic. And of course, like many other surfactants and other organic contaminants, they can degrade and form metabolites. These alcohol polyethoxylates, they can have different carbon chain length, and they can have different number of ethoxylate groups. And we selected 12 of them, and these are the 12 that we were analyzing. But before that, what we decided is to go to the field and then also take some soil samples. We visit 2 different locations with 2 different soil types, and we bring this soil to the lab to run 2 different types of experiments. As I say, for different carbon chain length and for different number of ethoxylate groups, we were running batch experiment to get absorption isotherms to characterize the absorption of this contaminant. But we do a reconstitution of the soil in the lab using this flow cell to do infiltration experiments. And of course, we were sampling water content, and we were sampling the concentration of this contaminant at different depths for the different 2 types of soil that I mentioned. In the case of this infiltration experiment, we were also using a flow and reactive transport model in order to characterize the absorption process, but under natural conditions, under hydrodynamic conditions. Here I show the goodness of our model for in the case of water content. Okay, we were very happy about how our model was able to capture the hydrodynamics of the flow in our flow cell for the 2 different types of soil. And then, as I say, for the different contaminants, we selected a total of 12 because we have 4 different carbon chain length and 3 different ethoxylate groups. Here I'm showing the result only for 1. As you can imagine, the number of isotherms, the number of flow cell experiments, it was increased exponentially. But here, just to simplify, I show the results, and we were also very happy about how our reactive transport model was able to capture the transport at the fate and transport of this non-ionic surfactant in our infiltration experiment. From this reactive transport model, we were able to get also the absorption coefficient, like the ones that we get, or the partition coefficient, like the ones that we get from batch experiments. And here is this main result from this intermediate scale. What we could see is that either if we plot by carbon chain length or by the number of ethoxylate groups, what we could see is that the absorption coefficient or partition coefficient that we get from the infiltration experiment was systematically smaller than the ones that we get from batch experiment. The batch experiment are in the x-axis, and what we have simulated thanks to the infiltration experiment and our reactive transport model is what I show here in the y-axis. In average, we get absorption coefficient that were 1.5 order of magnitude smaller than the ones that we get from batch experiment. So the general hypothesis and what is accepted in the community is that because under natural condition, you have less surface available than in a batch experiment. That's why you have less absorption. And that's why you need a smaller partition coefficient to capture the absorption process. And at this point, the question is if this hypothesis is true, and if it's the only reason of why the absorption coefficient that we get from or under natural condition is different than the ones that we get from batch experiments. So for that, we need to go one more scale down. And again, if we look, okay, we do a zooming into the soil and we see how a chemical, a contaminant, is transported through the soil. As you can see here, it can be a very complex process. This is an experimental image that we have done here in the lab. And then what we have here is in gray color are the grains. In blue color, we have the water. In black color, we have the air, like in soils, and that was a pulse of contaminant. Okay, we have the coupling of what is called multi-phase flow, the displacement of air and water, together with the transport of this contaminant. Here is just a dye, like an example, just fluorescent. And as you can see, of course, the contaminant is not in contact with all the grains, right? So this explains somehow the hypothesis that we have less surface available for absorption than in batch experiments. But let's go little by little, because what is relevant here is mixing. And what I mean by mixing is when we have two chemical species, or when we have a chemical species that we want that is absorbed, or that the main process or the main reaction is this surface reaction absorption, what we need is the collocation of this chemical together with the other one to produce the reaction, or the collocation of the chemical with the solid in order to produce the absorption. So this is actually what I mean by mixing. So two chemical species that are initially segregated, we have here two blobs, one in red color and another one in blue color, and this is what I mean by mixing. This is the key point, and this is actually the Trojan horse, I will say, that we are dealing, the people that we work on flow and transport in subsurface environments. But let's go in detail. What I show here is two experiments above what we call fully saturated conditions. This is what you can find in the aquifer. In the bottom, what we have is an experiment that contains also air in the pore space in black color. And this is what we can have basically in soil. So from the left to the right, what I show is just a zoom of this experiment, in which what we see is the plume of the contaminant, a pulse of a surfactant that is traveling from the left to the right through the porous medium. And what we can see here in the grains are the halos of the absorption. If we go to the unsaturated case, as I mentioned, the condition that you can have in soil, we can see also that the contaminant can be absorbed at the edges of the grains. So the first result that we get from this type of experiment, and again from different saturations, it's confirmed that when we have less water under natural condition instead of batch experiment, we have less absorption. And this is what we see here. We see how the mass is absorbed in time when this plume of surfactant is traveling through the porous medium. And at the end, we get the total mass that was absorbed, and we confirm the hypothesis that I mentioned before at intermediate scale. If we decrease the amount of water that we have in a natural soil, we get less absorption. But there is a lot of information in this region of this plot. Note that is log-log, okay, in which we can, looking in detail, we could see that is not the only reason. It's not the only reason that we have less absorption when we decrease the amount of soil, sorry, the amount of water in the soil. So the reason or another reason that explain why we have less absorption when we have less water in the soil is a faster dilution. And maybe you can think about, hey, Joaquin, if you have more water in the soil, you can dilute more. I agree. But what has been the main contribution of the group, and what it was very difficult to accept by our community, is that if you have less water, you dilute faster. I agree that you dilute more, but something is the magnitude and something different is the rate of the dilution. This is what I show in this figure. The mean concentration in these plumes versus time. When we go to long or longer times in our experiments, what we show here is that in the case of fully saturated condition, when we don't have air, at the end, the mean concentration in our plume of contaminant is decreasing diffusively. However, if we are in unsaturated condition, when we have air what we observe is that the mean concentration decreased faster. So this explains also why we have less absorption when we have less water in the soil. If you dilute faster, this is an isotherm. Okay, we have two Freilich and the linear. If you dilute faster, you also decrease the potential to absorb. And this is what we observe in this region of the plot that also explain why at the end I have less mass absorbed when we have more air or less water, as you prefer, in the soil. So just to conclude, because I have prepared a relatively short talk, at the end, the take-home message that I wanted to deliver in this presentation is that the current models or the existing models, they have problems to predict the chemical reaction in the subsurface. And the reason is because they ignore the incomplete mixing. They ignore the presence of chemical gradients. So in other words, if you see this figure that I show here for a dye in a natural soil, most of the model, we assume that below or above the front, everything is well mixed. But you agree with me that everything is not well mixed. All over these, the edges of these fingers, okay, we have chemical gradients. And it's there where the reaction happens. You need to have the chemical gradient, and you need to have the collocation of the two chemicals or the chemical together with the solid to have the chemical reaction. Just to conclude, here a beautiful video that has been recently produced in the group by one of the PhD students, by Isam Markale, in which we have been able to observe, in this case, a fluid-fluid reaction wave moving through a porous medium using magnetic resonance imaging. Here, the plume of reactant is cut by a vertical plane just to show half of the plume. And I show here only 3 grains or 3 bits, glass bits that we use to mimic our porous medium. Just to show you how the wave of reaction is moving through the porous medium and is deformed, and this deformation is the ones that produce the chemical gradients that at the end are the main control over the chemical reactions. And this work has been possible in the last years during this tenure track process, thanks to this group, which I thanks to them, because without them, all this work, all this and many other things were not possible. Thank you.